Welcome back to Chapter 9, Part 3. In Part 3, we're going to cover temperature effects, other environmental conditions, and the types of culture media used to grow bacteria. First, we'll look at temperature and microbial growth. By the end, you should be able to describe minimum, optimum, and maximum temperatures for growth, identify microbes based on their temperature ranges, and give examples for each category. All microbes grow within a temperature range defined by three key points: the minimum, optimum, and maximum temperatures. Growth is fastest at the optimum. Most human pathogens grow best at body temperature, around 37 degrees Celsius, and outside of their range, growth stops or cells die. Extremes will require special adaptations. We classify microbes by their preferred temperature ranges. Psychrophiles grow at 0 to 20 degrees Celsius, like those in the deep ocean or polar lakes. Psychotrophs grow best at around 4 to 25 degrees Celsius and can spoil refrigerated food, so our refrigerators are usually around 4 degrees Celsius. Mesophiles grow around body temperature, within the range 20 to 45 degrees Celsius, including most human microbiota and pathogens like E. coli, Salmonella, and Lactobacillus. Thermophiles grow at much warmer temperatures, between 50 to 80 degrees Celsius, like geobacillus that we might find in hot springs or compost piles. And geobacillus species are actually also endospore-producing bacteria. They're gram-positive and rod-shaped, very closely related to bacillus. Finally, we have hyperthermophiles, which grow between 80 to 110 or higher degrees Celsius, like pyrodictyum. This is a type of archaea that you would find in hydrothermal vents, kind of like the one shown here in this picture. This graph shows growth rate versus temperature, and notice how the curve, let's say this one, rises gradually to the optimum and then drops sharply. That steep drop reflects rapid protein denaturation past the optimum temperature. There's one missing here. The psychotrophs would be around here. They grow between 4 to 25 degrees Celsius, and they spoil our refrigerated foods. In cold temperatures, microbes can maintain fluid membranes with unsaturated lipids. These were fatty acid tails that have one or more double bonds, so they don't pack tightly and they stay fluid or liquid. They can also make antifreeze proteins. In the heat, they can use saturated lipids that do pack tightly to stabilize membranes. They can also make sure that their proteins and DNA have extra bonds for stability. This is the basis of heat-stable enzymes like Taq polymerase, which we'll talk more about, used in a procedure known as polymerase chain reaction. All right, for this checkpoint, bacterial human pathogens are generally mesophiles. They grow best between 20 to 45 degrees Celsius, with an optimum near 37 degrees Celsius, which is normal human body temperature. For the second one, thermophiles have higher guanine and cytosine content in their DNA. So it turns out G's and C's pair with 3 hydrogen bonds compared to adenine and thymine, which only form 2 hydrogen bonds. So these extra stabilizing bonds occur also in their proteins as well as these nucleic acids, and they help stabilize DNA at high temperatures, preventing it from denaturing. All right, let's recap 9.4. For the first objective, every microbe has a range of temperatures where it can grow. The minimum temperature is the lowest temperature where growth still occurs. The optimum temperature is the temperature where growth is fastest. So you'll see it has the steepest slope on a growth curve. And the maximum temperature is the highest temperature where growth is still possible, but outside this range, enzymes lose activity, membranes might change, and growth will stop. For the second one, I'm going to show you in the next slide using a table. So organized from coldest to warmest temperatures, remember psychrophiles are those microbes that could thrive in extremely cold environments, such as polar ice. Psychotrophs are also known as psychrotolerant organisms. These can grow in the cold, but have usually higher optima, so they don't usually grow primarily at the really cold temperatures, but halfway or a little bit on the higher end, although they can tolerate the colder temperatures, and these can spoil our refrigerated foods. Mesophiles were the ones that cause disease in humans and can also be found as part of our normal microbiota. Thermophiles prefer hot temperatures, such as compost piles and hot springs. And then hyperthermophiles prefer very hot temperatures, so extreme heat, such as in our hydrothermal vents. So for examples of each microorganism, we have the coldest psychrophiles. Examples could be Chlamydomonas nivalis, which are a type of algae that we might see in snow. And when we see this snow, we'll usually call it watermelon snow. It looks kind of pinkish. Another type of psychrophile are the Arctic marine bacteria. For the next category, psychrotrophs, we have Listeria monocytogenes. This is a foodborne pathogen. I see it a lot in the news. It can even contaminate frozen foods here and there. And we have Pseudomonas fluorescens. For mesophiles, these are pretty much anything found in the human body. E. coli, Staphylococcus aureus, Salmonella enterica. For thermophiles, we have one example. The source of Taq polymerase, an enzyme used in PCR, is Thermus aquaticus. That's where the name Taq, T-A-Q, comes from. We also have Geobacillus. These are also thermophiles. And then hyperthermophiles include Pyrodictium. This is a type of archaean, and we find them in the sea floor near those hydrothermal vents. Now we're going to move on to section 9.5. Here we're going to cover other environmental factors that influence microbial growth, things like salt, pressure, and light. We first covered osmotic pressure way back in chapter 3. We looked at how osmotic pressure affected cells. For instance, in a hypertonic solution, where there are more solutes in the environment than inside the cell, water is going to move towards the side with greater solutes, so it leaves the cell, and the cell membrane pulls away from the cell wall. This was known as plasmolysis. In hypotonic solutions, where there's a greater solute concentration inside the cell compared to the outside, water is going to enter the cell. And in cells with cell walls, the cell wall prevents the cell from bursting or lysing. So the cell wall can protect the cell from hypotonic conditions, right? The cell wall prevents lysis or bursting of the cell, but not much protection is available against high osmotic pressure where the environment is hypertonic. In this case, remember, water is going to follow its own concentration gradient and flow out of the cell. This results in plasmolysis and cell death. This explains why brines and layering meat or fish in salt are very good methods of preserving food, right? If there's a high concentration of salt outside, water will leave the cells, and these cells, like the microbes that might spoil the foods, will die. But some microbes actually thrive in high salt conditions. Halophiles need high salt conditions. So these organisms are found in marine environments where salt concentrations are around 3.5%. And this is in contrast to, for instance, human blood. Our blood salt concentration is about 0.9%. Extreme halophiles tolerate hypersaline conditions like the Dead Sea. And the Dead Sea is about 10 times saltier than the ocean. So percentage of sodium chloride is about 30%. Again, remember, compared to our human blood, which is about 0.9%. Halotolerant organisms like Staphylococcus aureus survive salt but don't require it. Overall, in all of these conditions, high salt or drying, because remember, water is going to leave the cells, lowers water activity, which means available moisture or available water that's not going to be present for regular cell activity, like enzyme function, transport or diffusion of certain nutrients, certain catabolic or anabolic pathways, or even our basic hydrolysis or condensation reactions. Some microbes adapt to high barometric pressure. Barometric pressure simply means the pressure exerted by the atmosphere or air at a given point. It's also called atmospheric pressure. Microorganisms that require high atmospheric pressure for growth are called barophiles, and these live in the deep ocean under extreme pressure. It's really difficult to retrieve intact specimens from deep in the ocean or reproduce their growth conditions in the lab so the characteristics of these microbes are pretty largely unknown. Switching gears and looking at light, other microbes can adapt to light. Photoautotrophs, we've seen before, use light for both energy and carbon fixation, while photoheterotrophs use light for energy but need organic carbon. And special pigments in these organisms allow absorption of light at different wavelengths. So earlier I mentioned the watermelon snow that you might have seen before if you've seen snow in Tahoe or something similar. Watermelon snow is caused by Chlamydomonas nivalis. This is a green algae that produces red carotenoids to protect themselves from UV light, from ultraviolet light. So this shows us how microbes can adapt simultaneously to intense sunlight and cold temperatures. These are combined environmental pressures. All right, so what photosynthetic pigments were described? I just mentioned carotenoids briefly. These are an orange-red pigment generated by Chlamydomonas that generates that watermelon snow. There's another one in the book I didn't talk about called Bacterial rhodopsin. It's a pigment similar to retinal in our eyes, and they use it for light-driven proton pumping. So don't worry about that since I didn't talk about it earlier. These pigments allow these microbes to harvest light energy even though they're not chlorophyll-based like plants or cyanobacteria. For the second one, fundamental stress of a hypersaline environment of a cell. The main stress is osmotic stress caused by the really high salt concentration outside of the cell. Without adaptations, water would move out of the cell, causing that plasmolysis, that dehydration and shrinking of the cytoplasm and cell death. So halophiles can adapt by accumulating compatible solutes, so they're not constantly losing water. There are other adaptations too, but they're going to be beyond the scope of this section for now. To recap 9.5, I'm going to show you a table on the next slide that summarizes both of these objectives. So in this section, we talked about barophiles that really like high atmospheric or hydrostatic pressures. So these might live in the deep ocean, and they have adaptations to resist this high pressure in their membranes and proteins. Sometimes instead of the term barophile, you might see other texts use the term piezo-philes. That's another name for the same type of microbe. An example could be Coelia. These are deep sea bacteria. We also talked about halophiles. These like or prefer environments with high salt concentrations. So these use compatible solutes or a method I didn't talk about where they can retain potassium inside of the cells to balance osmotic pressure. So one example are these Archaea, halobacterium, that like, again, high salt concentration environments. We also briefly talked about phototrophs again. These require light for energy, and they have pigments such as chlorophylls or other pigments such as bacterial rhodopsin to harvest light energy from the sun. Examples include cyanobacteria or purple sulfur bacteria. Finally, in section 9.6, we'll review different types of media that are used to grow bacteria. For our media types, the most general type is the one up here, general or all-purpose media, that supports the growth of many different types of bacteria. So one common example is tryptic soy broth, which is just like the tryptic soy agar we've been using in the lab. TSA is actually just the solid form of the same medium, TSB. We just add some agar to it. Nutrient broth is actually also a type of general or all-purpose media, but it's also in this category. So you'll see this is true for a lot of these media. They fall into multiple categories. Nutrient broth is also complex because we don't know exactly what's in it. And this is because we're using some kind of extract from living organisms, such as yeast, plants, or meat. Defined media is the opposite, where you do know everything that went into your medium. When we're making defined media, this is usually through a recipe, and usually you'll add some amino acids, different types of carbohydrates, different lipids, etc. And you mix everything together with water, but you know everything that's in it compared to complex media where you don't know the exact composition because of the extract that's being used. Enriched media is when you add specific growth factors, vitamins, or other specific nutrients, And this is used when you're growing fastidious organisms. These are organisms that only grow with specific nutritional needs. So when you meet their specific nutritional needs, we often call them picky eaters. And these types of media can be used to culture a pretty wide range of microbes. Although it is true that some microbes cannot be cultured in the laboratory, and we only know they exist or we can identify them based on their DNA or RNA sequence. We also have selective and differential media. In selective media, this will inhibit a certain group of organisms and promote the growth of others. In differential media, we're going to see metabolic differences between microbes. So selective media inhibits unwanted species. For instance, we've already worked with phenyl ethyl alcohol agar, and we saw that it inhibits gram-negative organisms. McConkey agar, which we're going to work with in the future when we start studying gastrointestinal bacteria, we're going to see that McConkey selects for gram-negative bacteria. So it does the opposite of PEA. Differential media shows metabolic differences between organisms, and this is usually in the form of some kind of color change. So one example is phenol red broth. For instance, phenol red lactose broth, which is red to start with, and if lactose fermentation occurs, the tube will turn yellow. Some media will combine selective and differential properties. For instance, I didn't talk about it yet, but McConkey is actually a type of agar or broth that actually is both selective and differential. I'll show you an example in a bit. And then finally, we have enrichment cultures that repeatedly favor a target organism. So enrichment cultures will foster the preferential growth of a desired microorganism that's only going to represent a fraction of the organisms present in an inoculum, in a sample. So these cultures will enrich or increase the target organism's numbers without necessarily inhibiting the others. One will grow better, and over time you'll have most of that preferred one. So here's an example of that phenyl ethyl alcohol agar example from lab. Here on the left we have a tryptic soy agar plate where everything pretty much grows on it. This is a general all-purpose medium. On the right we have phenyl ethyl alcohol agar, and I have two organisms growing on the plates. On the right I have Staphylococcus aureus. This is a gram-positive organism. And on the right it's also S. aureus. So it's growing well, this gram-positive organism. On the left of both plates, I have a gram-negative organism, Enterobacter aerogenes, and you can see on the PEA plate, it's being inhibited. So PEA inhibits gram-negative bacteria and allows for the growth of gram-positive. For differential media, we've seen phenol red broths. Here's an uninoculated control tube. Let's pretend it's lactose. If your bacterium ferments lactose, you'll produce acid, and that will turn phenol red to a yellow color, since phenol red is yellow when acidic, and we saw fermentation can occur with gas or without gas. And I talked about McConkey agar as a selective medium, but it's actually both selective and differential. McConkey contains bile salts and crystal violet, and those two are going to inhibit gram-positive bacteria. So there are actually two gram-positive species plated on the left and right that you don't see any growth on because they were inhibited by the bile salts and crystal violet. And these two that did grow, these are both gram-negative. So these are gram-negative organisms. And I also can see color differences between the two. So it turns out that there is neutral red as a pH indicator in this agar, and when lactose fermentation occurs, this pinkish color is generated. And the bottom, there is no lactose fermentation, so it generates this yellow color. So you have to be careful. Depending on the pH indicator you use, the color changes will be different. In the broth that I just showed you, phenol red broth, yellow indicates acid and fermentation. But in McConkey agar, it's pink, this pinkish red color that indicates fermentation has occurred. Another medium we're going to be working with in the future is also selective and differential, and this is called mannitol salt agar. The salt portion of MSA, mannitol salt agar, is the selective ingredient because only bacteria that can tolerate salty conditions or environments will grow. And MSA has about 7.5% sodium chloride compared to human blood, for instance, which was 0.9%. So it's pretty salty. It also contains a differential ingredient. This is a carbohydrate, mannitol. So mannitol might be fermented by your microbe, and if it is, the medium will turn yellow. And this is because there is a pH indicator in here called phenol red, just like our phenol red broths. So here there are two organisms growing. So I know both of these must be salt tolerant because they grew pretty well. But only the top one ferments mannitol. So acid was produced and the phenol red turned yellow. Here there's no yellow, so mannitol fermentation did not occur. All right, for this checkpoint, we have to distinguish complex versus chemically defined media. In complex media, the chemical composition is not known because it contains digest or extracts from plants or animals or other microbes. This provides a really rich and broad mix of nutrients and supports the growth of many different types of organisms, including even sometimes those really picky ones, those fastidious ones. So examples of complex media include nutrient broth. Tryptic soy broth or tryptic soy agar is actually also complex. And then something else we're going to work with in the future, which is called blood agar. And blood agar is just TSA with 5% sheep's blood. Chemically defined media is when you have the exact composition. You know the exact composition. Every chemical, every concentration of those chemicals is known. So this type of medium only contains specific chemicals that's chosen by the scientist. And we usually use this to study very specific nutritional requirements. For the second bullet point, selective versus enrichment media. Selective media contains ingredients like different types of dyes or salts or even antibiotics that inhibit unwanted microbes while allowing the target microorganisms to grow. So for example, McConkey agar selects for the growth of gram-negative bacteria by inhibiting gram-positive bacteria. In enrichment media, these contain conditions or nutrients that favor the rapid growth of a specific desired organism. And this is from a mixed sample without necessarily inhibiting others. So one of those organisms will grow much better in the medium. I also thought a chart would work best to recap this section, so let's go over that in the next slide. All right, so we had general or all-purpose media like TSA. This supports the growth of many different types of microbes. Enriched media had those extra nutrients to support the growth of those picky, fastidious organisms, such as blood agar. Selective inhibits certain unwanted bacteria while allowing the other target groups to grow, such as McConkey or MSA. Differential media shows you a metabolic difference, usually through a color change. We also saw that in McConkey agar. We're going to see it in the future in blood agar, and I also mentioned phenol red broth. Defined media is when you know every single ingredient in that medium. And in the future, we're going to work with something called minimal salt medium. And enrichment media. This is when you have a mixed culture or a mixed sample, and the medium has ingredients that encourages the specific growth of one of those organisms. And we'll talk about these more in the future. And that takes us to the end of Chapter 9. In Chapter 10, we're going to look at the biochemistry of the genome, as well as the structure and function of DNA and RNA. Thanks for watching.